do, I always do chapter 6 before chapter 5.
Makes sense.
And next week in lab, you're going to start identifying your unknown bacteria. And one of the things you're going to have to do to identify your bacteria is grow it on different types of special media. Media that has special nutrients in it, and also special dyes in it to serve as an indicator when the nutrient is used. So all of that information is in chapter 6. By the way, in the movie they mentioned a micron. Do you know what a micron is? We'll do it when we do our metric conversion. Micron is just a simplified way of saying micrometer or micrometer, which is equal to 10 to the negative 6 of a meter. So we'll do that when we... I have these PowerPoints and there's so much different stuff in it. It's kind of crazy. One of these days I need to break them apart. So your textbook just has so much stuff in it. All right, this is a test you're going to do in lab next week. And this figure you've seen already when we did classification. Just want to make sure, you know, autotrophs from heterotrophs. This explains that. This is hydrogen peroxide being broken down to water and hydroxyl. Right. Remember this? Oxygen concentrations. Oh, you need my little cheat sheet. That's one of the things on your second test. We'll have questions about oxygen requirements. There's also a page in your handouts package that talks about oxygen requirements. So if an organism is obligate, it must have a certain set of conditions. So if it's an obligate aerobe, it's obligated to have air, or O2 gas. Not elemental oxygen, but O2. If it's an obligate anaerobe, it's obligated not to have oxygen. And in fact, oxygen may kill it. Facultative probably prefers oxygen, grows better in oxygen, however, can still survive without oxygen. And air-tolerant anaerobes, they actually live using some other element instead of oxygen. So if oxygen is available, they just don't use it. It doesn't kill them, they just don't use it. The reason I brought this up again is because eventually you're going to be doing some experiments where you grow bacteria on the surface of your Petri plate and do other Petri plates where the bacteria grow throughout the agar. And what you'll notice is the bacteria, if they're facultative anaerobes, which all of your unknowns are going to be. If it's a facultative anaerobe, it means it can live with oxygen, but probably does better with oxygen. Can live without oxygen, but doesn't do as well. So if you have bacteria living throughout the agar, the bacteria that are on the surface are going to make bigger colonies. They're going to grow better than the ones in the agar. The ones in the agar, they're going to have smaller colonies. And that will be one of your quiz questions when we get to that. But I just didn't want you to forget the terminology and all that stuff. Temperature, we've already figured it out. There's an optimal temperature. A lot of bacteria actually like human body temperature. That's called mesophilic. Meso is middle, philic is love. Human body temperature is mesophilic bacteria. So bacteria consider the human body the middle range. If it's psychrophilic, that means it likes it really cold, like in your refrigerator. Or if it's one of those archaea bacteria, maybe even in the polar ice caps. Like, can you imagine that crap? Look, and it's red bacteria. Look, it's growing on freaking ice. And here's ice crystals, and here's the bacteria growing.
So
the ice
that's
crystals.
what makes the snow dirty in spring.
No, that's dog pee. We probably don't have cytophilic bacteria in the snow here. Oh, but we're from, maybe where you live, maybe where you came from, but not in Jersey. It's just dirt from the streets and stuff that makes it so dirty in the spring.
Pretty much.
But there are some bacteria that can live with it back home. And thermophilic likes heat, hotter than human body temperature. So these slides are basically self-explanatory. I don't know, the way your textbook goes, I apologize. If you read the entire textbook, page after page after page, probably seems kind of organized, but put so many different things into the same thing. So for some reason in that chapter 6, he also puts the biofilm development. But we talked a little bit about biofilm and why biofilms are important. Bacteria can actually communicate, quorum sensing, and as they communicate in these biofilms, they can cause more trouble. They can be resistant to antibiotics, and they can protect other microbes behind the wall. So it's weird, all of a sudden the biofilm comes up in there. And then remember, these are shapes and arrangements of your bacteria. What bacterium do you think this one is? Going by the red pigment alone, I would say Serratia marcescens. Yep. Okay, and this is the same thing, just that's on the, so your textbook has a tendency to do that. Give me one figure with both pictures and then separate them. So, streak plate method, you already know. This is the quadrant streak plate method, 1, 2, 3, 4 quadrants. In our lab PowerPoints, it showed us to do 3 quadrants, but then they still call them quadrants. I think that's totally wrong. To me, a quadrant is a quadrant, and I would call that whatever a tri-streak plate method, but whatever. You know how to do streak plate? And you know why you do streak plate method? Because you're trying to essentially spread them out or dilute them so you get these separate colonies. All right, we're going to do later in lab the pour plate method, but it's in chapter 6, so I just left it in chapter 6. But we're actually going to do a pour plate method later in lab. It's the pour plate method where you get colonies on the surface and colonies down in the agar. Because you're going to mix your bacteria into the agar, and then you're going to pour it in the plate. Whereas what we've done before with streak plate and everything is we had our agar and we put our bacteria on top. There's another method called spread plate, where you take your bacteria in liquid and you spread them over the surface of the agar. So we'll do that in lab. Now, this is just showing you a different way. You saw it in the aseptic technique video. A lot of microbiologists use what's called a slant. So they pour the molten agar in the test tubes, and then they actually let them solidify at an angle. And the reason they do it is so you get a much greater surface area to grow your bacteria on. And selective media, we're going to work with next week. Selective medium means it's got ingredients in it that help one kind of organism grow and inhibit another kind of organism. So as you can see, at a higher pH, is higher pH more basic or more acidic? More basic. And 7.3 is actually neutral, like our tissue fluids. So neutrophilic bacteria are bacteria that like human body tissue fluid pH, which is 7 to 7.4. So 7 is neutral pH. pH scale goes up to 14, 0 to 14. So if your pH is more acidic, that's when the fungi like to grow. And actually, there's a little clip about, you know when you get a vaginal yeast infection? Actually have the pH for that, if I can find it.
It should be the parasitic inside the clean.
Well, the lactobacillus bacteria, remember, they like it acidic. And fungi usually like it a little more acidic than bacteria do. But I have the exact figures because I volunteered at a women's health place. And what they would do, and what they would do is literally take, you know that pH paper they use in chem class?
Mm-hmm.
They take a little strip of pH paper and touch it to the whatever fluids you had down there, and then read the pH to determine if it was a bacterial vaginosis or a yeast vaginitis. Normal vaginal pH is 3.5 to 4.5. So that's kind of acidic. Yeah. And the lactobacillus acidophilus bacteria that live there naturally help keep it that way. If your pH in your vagina gets more neutral, up to 7, that's when it allows Candida to flourish. So Candida actually likes kind of a neutral pH. And if you douching neutralizes the pH. And that's why sometimes people think, oh, I got to douche to get rid of this thing, and you're actually making it worse.
What did you say the natural pH was?
3.5. And then
That's what I meant, acidic pH.
Normal
The
is
normal.
3.5 to 4.5.
That's normal.
The yeast is neutral, 7.0, or in that range, probably 6.9 to 7.1 or whatever, you know. There's probably a range.
Stupid question, maybe.
Yeah.
What do you mean by douche?
Squirt water up there, well, not water, but chemicals.
Okay, I like it.
That's what you use a douche